Well, good morning, students. This lecture is the third in the series of the lectures on genetics, and the title of this lecture is Human Chromosomes. Now, as in the last lecture, I said that when Walter S. Sutton gave the chromosomal theory of heredity, and he said that the genes are the factors which determine the characters are located on chromosomes. After that, okay, this was in 1902. After that, the scientists throughout the world who were working in biology, plant biology or animal biology, okay, they become interested in chromosomes, okay. So this lecture is based on the chromosomes, okay. We will learn about the human chromosome in this lecture. Okay, I go to the next slide. And now, chromosomes are present in nucleus, okay? We very well know that chromosomes are present in nucleus in the form of strands. Strands means a thread-like structure. Strand means thread-like structure. And these thread-like structures which are present, a bunch of the thread, okay? Network of the thread, okay? Which is present within the nucleus. This is called as chromatin. And why it is called as chromatin? Because you can stain this bunch of the thread, okay? These threads which are uh, forming a network can be stained, okay, with the hematoxylin or any basic dyes, and it looks blue. So this is called as chromatin. Now this chromatin material which is present in the nucleus, okay, at certain places it is darkly stained, at other places it is lightly stained. So this chromatin network which is in the nucleus, at certain places it is light and at certain places it is dark. Where it is light, this portion of the chromatin is called as euchromatin, E-U-C. Chromatin, that is euchromatin. And the places where within the nucleus this chromatin material take the dark stand, it is called as heterochromatin. It is called as heterochromatin. So there are two kind of the chromatin present in nucleus when you observe it under the microscope, in especially in eye immersion lens, okay, at high magnification, okay, in a nucleus which is resting nucleus, okay, which is interphase nucleus, it is called as resting nucleus, you will get heterochromatin and the euchromatin, okay. Now the chemical constituent of this chromatin, that means it is made up of the DNA, deoxyribonucleic acid, okay, DNA, it is made up of the DNA. Now during the cell division, now this chromatin material further condenses, okay. There are heterochromatin patches are though present in a resting cell, but when the cell division is taking place, okay, this chromatin material become spatially condensed, okay. Hmm, and it forms chromosomes. That means the chromatin thread-like network becomes so condensed that rod-shaped structure begins to begins to seen in the during the cell division. Okay, so what are the chromosomes? Chromosomes are nothing but the rod-shaped structure which can be only seen during the cell division when a cell is dividing, and this has formed, the chromosomes have formed due to the tightly condensation of the chromatin material. What does it mean? Okay, it means that the chromosomes, they are mostly in the interphase or resting nucleus, they are not tightly coiled, okay, they are in expanded form, loose form, unbound, okay. and that is the reason why we don't see the chromosome all the time in a nucleus. We see the chromosome only during the cell division when they are especially very tightly coiled. Okay, so the chromosomes are visible as rod-shaped structure only during mitosis or meiosis. Okay, any kind of cell division. After the cell division is over, these chromosomes, which were rod-shaped structure, dense, okay, tightly coiled chromatin material, they again uncoil, okay, for the interphase, okay, or for the resting phase of the cell, they become uncoiled to form the chromatin. Okay, so now they are no longer called as chromosome, and the uncoiled chromosome is now called as chromatin. Okay, chromatin. So please differentiate what is chromatin and what is chromosome. Okay. Now the question comes, why euchromatin is lightly stained and heterochromatin is darkly stained. Okay. Now I will explain you. The euchromatin is lightly stained because most of the part of the chromatin material is open up. That is, the DNA strand is uncoiled at that time. Okay, most of the portion of the chromatin material is not tightly coiled. It is said to be open. Okay, or open phase chromatin. Okay, and this is the reason why it is not darkly stained with the basic dyes like hematoxylin. Okay, it looks light. It looks light. And then question comes, why it is not coiled? Okay, now most of the portions of chromatin material which are not tightly coiled, okay, they contain genes which is under the, I mean to say, active form. Okay, so the genes are there, structural genes or any other kind of genes are there, which biochemistry people will tell you many kind of genes are present. A structural gene is a gene which is responsible for the coding for the synthesis of a protein enzymes, okay, etc. Now, so the gene is active, and that's why it has to uncoil. A tightly coiled gene in a DNA strand cannot transmit the codes in the form of the messenger RNA. Hence, it has to open up. Hence, and that is the reason why it is lightly stained. Okay. While in case of heterochromatin, okay, DNA strand is of the various chromosomes, they are tightly coiled, okay, because at that time the I mean, say the genes are not active. I hope it is clear. Now, let us come to the morphology or structure of a chromosome. Once we have understood what is chromatin and what is chromosome, and chromosomes are visible only during cell division, okay, within the nucleus. Let us see the structure of chromosome. There are 46 chromosomes, and this 46 chromosome number in a somatic cell. Okay, what are somatic cell? All the cells of body except the gametes, that is sperm and ovum. Okay, all other cells, they are called as somatic cells. They contain 46 number of chromosome, and this number is called as diploid number, and it is represented by 2n. It is represented by 2 because it is the double set. It indicate because in the gametes it is in single set. Okay, that's why it is called as 2n. Otherwise there is no reason to represent it as 2n. Now this, out of this 46 chromosome, there are 44 autosomes and 2 sex chromosome. Okay, autosomes means those chromosomes on which genes are present for all the characters of body except the sex. Okay, except one which is related to the sex. Okay, or determination of sex, or related to the secondary sexual characteristic, related to the reproduction. All other genes are located on this 40. So they don't perform the function in a very broad sense. I am saying they don't perform any function related to the reproduction or the, I mean to say, uh, 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 gamete formation, CFAR. So there are two sex chromosomes. In female they are XX and in male XY. Okay, that you know. Now what are 44 autosomes? And these 44 autosomes, they are present in a somatic cell in 22 pair. That means one pair consists of two chromosomes, and these two chromosomes of a pair are exactly the same. Same means the in structure they are the same. Their length, their width, and their position of centromere, okay, which is almost exactly the same. And that's why the members of a pair are called as homologous. So homologous pair of chromosomes. So there are 22 homologous pair of chromosomes, out of which, okay, these 22 pairs, okay, the 22 have come from mother and 22 have come from father, and thus they have formed the pairing. Okay, now then there are two sex chromosomes and they are called as X and Y. In female there are 44 autosomes and two X chromosome or sex chromosomes, right? And in males there are 44 autosomes, one X and one Y chromosome, that you already know. Okay, now the size, if you see the size, average size, okay, average size of a chromosome out of these 44, it is 5 millimicron, 5 mu, okay, 5 millimicron. Okay, you know what is a millimicron. It is 1 upon 1000 part of 1 millimeter. Okay, is 1 millimicron. Very, you cannot see. You cannot see it with naked eye. You have to use a high power. That means microscope. Not even it is visible in low power. Okay, all will measure only half a millimeter. Okay, when you will arrange this I will say chromosome 44 touching to each other in a single row, this will measure only less than half a millimeter, less than half a millimeter, not even 1 millimeter, which is hardly visible with the naked eye. Okay, but when you will extend this DNA strand of this chromosome, okay, uncoiled it and then touch, I mean say then arrange them in a row of all 44 chromosomes, 46 chromosomes, then this will measure few meters in length. See, on one side it is few meters and other side it is less than half a millimeter. Okay, so this indicates the tight coiling of these DNA strands within the chromosomes. Okay, now sperms and ovum, they contain only 23 chromosomes, and half the number of chromosomes are known as haploid. So remember haploid and diploid terms. Okay, I am introducing you these two new terms, and this is represented by n. Whenever it is written as n, it means half the number of chromosome and it is called as haploid. Okay, let's go to the next slide. Now we come to the shape of the, I have just roughly said that the shape of the chromosome is rod shaped. Okay, it is rod shaped. But this shape of the chromosome, which is rod shaped, okay, is present only during the mitosis. If you see a cell cycle here, let us see the cell cycle, different phases of cell cycle. See, this is the different phase of the cell cycle, right? Cell cycle is shown here. The whole of this from here where my pointer, this whole large area in a cell cycle is of the interphase, means where the cell is almost resting, okay, or preparing itself for the mitosis. And then here m thing is written here, and this is the phase where actual the cell division is taking place and two daughter cells will result. Let us see in the G1 phase of the cell cycle. Now chromosomes are not visible in microscope, with any microscope, okay, because they are in extended condition. Okay, so here the DNA, which is double helix strand, okay, it is an extended condition. Okay, it is quite extended in condition. And here what you see is the now this material, DNA material, these two strands will just they will go away from each other and a new strand of DNA will form, what is called as replication of DNA. This takes place in S phase of the interphase, S phase of the cell cycle. Now, then because of this double DNA material has formed here, then this double DNA material, one which was old and another which was new, they will start chiling as it was there in the beginning of G1 phase. Okay. Now this will be, these two strands now will attach at the constriction called as centromere. So they will further chyle. Okay, further condensation will take place. And when the metaphase will approach, this condensation of the strand of the which are now called as chromatids. So it is a single strand is called as chromatid. So in a chromosome there are two chromatids. Okay, these two chromatids will form and it will become condensed, and now it is visible as a uh, I mean to say chromosome. Now it is under the microscope, high power microscope, you will see that this condensed structure with a centromere and two chromatid rod shaped structure attached to centromere, that is now called as the chromosomes. Okay, otherwise this chromosome was not visible. And then when the mitosis is taking place, okay, in the last terminal stage, these two chromatid, due to the longitudinal splitting into the centromere or primary constriction, these two chromatid will go to the opposite pole in the daughter cells and cell division will be completed. So what I mean to say here is that the chromosomes are only visible during the cell division, whether it is mitosis or meiosis, immaterial. Okay. Now, after learning about the shape, let us come to the structure of the human chromosome. Okay, the metaphase chromosome consists of the two chromatids. Okay, see here, which I have already told you. Okay, when during the cell division, if you will see a chromosome, it consists of the two chromatid. Okay, and I told you how two chromatid has resulted due to formation of the new DNA material. Okay, against the existing DNA strand. Now two chromatids are attached as centromeres. See, this one rod is one chromatid, this another rod is another, and these two rods are connected as the uh, centromere, which is called as the primary constriction. This is called as centromere or primary. Now this centromere divides chromosome in short and the long arm. Okay, the centromere may be slightly placed away from the midpoint, and then one arm will be long, another arm will be short. The short arm will maybe called as P arm, small letter of English. Okay, and long or the Q arm. Okay, I will tell you further the significance of P and Q. Okay, now sometime at the end or in the middle and another constriction beside this primary constriction or centromere, and other constriction in this rod shaped chromatid may be seen, and when it is seen very close to the tip, this is called as, otherwise also, sorry, if suppose constriction is here where my pointer is moving, this will be also called as secondary, because it is primary, this will be called as secondary constriction. Now if it is near the tip, okay, secondary constriction, so whatsoever uh, remaining part at the tip of the chromosome is there, that is called as satellite. Okay, it is called as satellite. And here the secondary constriction and satellite, they are involved in the RNA. Okay, formation of the RNA, which is another subject. Okay, now we move further. Now let us come to the classification of chromosomes. Okay, chromosomes are classified in various ways. and this is the first classification based on the position of centromere. You see this is the centromere here, where it is close to the center, where it is away from the center, it may be still away, okay, and it's at the tip, okay, it may be at the tip. Right. So based on the position of centromere, okay, they are also based on the presence or absence of satellite, okay, they are classified like that, where very close to middle it is metacentric, then submetacentric, acrocentric, and telocentric. When it is, there is no long and short arm, only one arm is there, okay, that is called as telocentric chromosome. So this was the one of the simplest and the earlier classification of chromosome. I am moving to the next classification of chromosome, and this is fairly known classification of chromosome, you must have already studied. This is called as the standard or Danver classification, and in this chromosomes are arranged in descending order from longest to the shortest, and in this way there are A to G group. So A group consists of 1 to 3 chromosome, B consists of 4 and 5 number chromosome, and 6 to 12 are just classified in C group, and then I mean say put into the C group, 13 to 15 in D, and 16 to 18 in E, and there are 19 and 20 in F, and the very short chromosome 21 and 22 are in G group. But then here you are seeing the two more chromosomes, and these are the sex chromosome. If it would have been a female, there would have been 2X. Since it is a male, it is X and Y. Now, this X and Y belong to different groups. For example, this X chromosome belong to C group, okay, according to its length, and the Y chromosome belong to the last group, that is the G group. This is a standard classification, and when karyotyping or the chromosomes are prepared in laboratory of an individual, they are cut and put as per their descending length, okay, and this group is formed. This is called as karyotyping. Let's go to the third classification, and this is known as nomenclature. With the advanced techniques, okay, when came, then we could stain the chromosome which was showing various pattern, I mean say bands, okay. These banding patterns was seen here. See, this light and dark band were seen, and these banding patterns were was the specific characteristic of an individual chromosome. That means a homologous pair will show the same banding character, but it will be different than the other chromosomes, okay. So this banding pattern, when after staining the bands were obtained, this become helpful in identification of chromosome, and then this banding pattern, they were given the nomenclature like that. There may be, they were divided into zone, and this zone were starting from centromeres and were going towards the tip in the P arm and the Q arm also, that is long arm and short arm. So this is first zone, this is our second region or zones, like that, first, second, and third. An individual region was having many bands, and they were given the numbers, okay, they are given the number. So in this way one can be very specifically tell that this in zone number 2 of the long arm of a chromosome at position 2, okay, in zone or region 2 at position 2 there is some defect, so you can pinpoint the defect present in a chromosome with the help of this banding pattern, okay. But I will stop here with the nomenclature. There are many more advanced techniques for the staining of the chromosome, which we will deal in the next lecture, okay. So this I think is the last slide, which have told you about the size, shape, and nomenclature and classification of the human chromosomes, okay. Thank you very much.